Hello and welcome to Mr. Yates's AQA Biology tuition videos. Today we're going to be looking at something that occurs in the first year of the new A level under the first topic of biological molecules, and that is going to be carbohydrates. So we're going to look at 5 main areas today. We're going to look at what a carbohydrate is, how to define it. We're going to look at the chemical structure of glucose. We're going to look at making and breaking bonds. We're going to take a look at other carbohydrates that we find, and then lastly we're going to look at how do you test for carbohydrates. So, what is a carbohydrate then? Well, to define it, you just have to look at the name, carbohydrate. Okay, what does that mean it contains? Well, it obviously contains carbon, it contains hydrogen, and it also contains oxygen. And all carbohydrates have these in some sort of proportion or other. We can generally split carbs into sort of 3 main groups. Okay, we can have one group over here, which are the polysaccharides. Polysaccharides, so that would be things like cellulose, glycogen. Okay, and then we can come this way to our sugars. We can split these up into 2 little subgroups. So those are monosaccharides, which would be glucose, fructose, and you have your disaccharides, which would be sucrose, maltose. Okay, so now on to looking at probably one of the most basic carbohydrates that you can get, and that is a glucose molecule. And here we have one over here. You have to know how to draw this. No easy way around it. You just got to sit down, you've got to draw it out, you've got to learn, you've got to get it in your head. Okay, so this is actually a molecule of alpha glucose. Alpha glucose. You can also get beta glucose, which makes up cellulose over here. But you don't really have to know how to draw that. Might be worth learning it. It's not a massive difference at all to alpha glucose. So easiest way I think is obviously you've got to count your elements basically. I always start with your oxygen in the top right. Okay, you should know that oxygen has got 2 valencies it needs to fill. So we go down there and we go along here. You're essentially drawing a hexagon. Okay, with each corner except the top right being a carbon. So we're going to go around here. And now we've obviously got 5 carbons. Glucose is a 6-carbon molecule. The last one stands at the top here, attached to H2OH up there. Okay, now carbon has got 4 valencies to fill, so we have to fill them up. We've got this empty one down here, we fill with a hydrogen. We've got these 2 down here. We have a hydroxide, a hydrogen, and a hydroxide. And then at this side we've got a hydrogen, hydroxide, and facing the same way is a hydrogen and a hydroxide. The only difference between this molecule of alpha glucose and the molecule of beta glucose is beta glucose does have these 2 switched around. So the OH is at the top and the H is at the bottom. We'll have a look at why that is important a little bit later on. So now we're going to have a look at how do we form a disaccharide. Di meaning 2, saccharide meaning sugar. So we have 2 glucose molecules there. I've not drawn all the elements, but you can see we have the subgroups of the H and the OH on each one. So what we're going to do is we're going to form this disaccharide from 2 glucose molecules, and we're going to form a molecule of maltose. That is what you form when 2 glucose molecules are joined together. Now that reaction that happens is called a condensation reaction. A condensation reaction. Again, think about what that might involve. Condensation. You get it on your windows. It's water. Okay, so there's going to be water involved somewhere. Okay, and even if you don't know anything about this, you can probably, by process of elimination, where you're going to get water from by bonding this molecule together. Well, water is obviously H2O. Where are 2 hydrogens and oxygen? Well, hello, here they are. Okay, so what happens is these molecules will form water, And what you get is 2 molecules of glucose forming here with this bond in the middle. Now, this bond is known as a, if I can spell it, a glycosidic bond. Glycosidic bond. This is a fairly strong bond, okay, between these 2 molecules of glucose. What about then breaking this apart? Okay, so breaking bonds, not breaking bad, breaking bonds. Okay, so we'll draw this again down here. We have the molecule of maltose. Okay, there it is with the glycosidic bond in the middle. So the reaction now that happens is known as a hydrolysis reaction. Hydro meaning water, lysis meaning being broken down by. So hydrolysis, broken down by water. Okay, so we add water into the mix, into here, H2O, and hey presto, we now get 2 molecules of glucose, just like we started with. So let's have a look at some common disaccharides that you find. Well, I've already mentioned that glucose plus glucose will give you maltose. So what about some other simple disaccharides? Well, if you take glucose and you bond it with fructose, you get something called sucrose. And also if you take glucose and you combine it with galactose, you get lactose, a milk sugar. So now let's have a look at some common polysaccharides. Poly, many, saccharide, sugar. Okay, so these are just long units of repeating molecules, polymer, repeating units. So the first one we're going to have a look at is the molecule starch. Okay, starch is the plant storage molecule. It's insoluble, which makes it very good because it doesn't affect the water potential. It doesn't change the water potential of the cells, so cells therefore are not taking up water by osmosis. It's not pure, meaning that it is a mixture of 2 submolecules, those being amylose and one called amylopectin. So amylose then is it's a long glucose chain. It coils up into a helix, a little bit like that, and in between then it's held by hydrogen bonds. Okay, so amylopectin then is very branched. It's a very branched molecule. Okay, so it's got lots more ends. It can be broken down very quickly, or a lot more quickly compared to amylose. So broken quickly. They're both broken down by amylase into maltose, but both at different rates. Obviously, amylose takes a lot longer as it's essentially one long molecule, whereas amylopectin shorter time because of its branched nature. So the next common polysaccharide, sticking with plants, is cellulose. And as I said before, cellulose is made up of beta glucose. So how is beta glucose different? Well, a molecule of beta glucose, I won't draw it all out, it's very similar. HOH, but it has the OH and the H different here, as I mentioned earlier. So what does that mean in terms of its structure? Well, if you think about cellulose uses, cellulose are found in cell walls. Cell walls need to be strong, they need to be rigid. So when we connect lots of molecules of beta glucose, there's this molecule of hydrogen down here. Now hydrogen can then bond to the hydrogen on the next molecule here, and what you end up with is a long lattice-like chain, so a bit like a brick wall really. So I'll just draw those. Imagine the long lines are the cellulose molecules. These bonds in between here are the hydrogen bonds. Okay, just like a brick wall. Very strong, very rigid, and it makes it ideal for plant cell walls. We can't digest cellulose because this sort of glycosidic bond that forms in beta glucose cannot be broken down by amylase, but it requires a specific cellulase enzyme, and only sort of bacteria. So bacteria found in sort of herbivorous animals in their guts. That's the only thing on the planet that can break down cellulose. So these animals have got this mutualistic bacteria in their guts, so therefore they can digest it. We can't, and we refer to that plant matter as fibre. Essentially helps us evacuate our bowels sufficiently. So last molecule then we're going to take a look at is glycogen. And this is the animal storage polysaccharide. You mainly find it in muscle cells and in the liver. Pretty similar to amylopectin, so it's very branched, so you've got this branched-like structure here. Okay, it can be broken down quickly, which is good if you're running low on blood glucose. Actually gets broken down to glucose by an enzyme, glycogen phosphorylase, but you don't have to know that. So that is glycogen. And finally, we'll take a look at how to test for carbohydrates. So we'll take the simple one first, and that is the starch test. So, test for starch, all you do is you add iodine, and if starch is present, it will go blue-black. So very, very easy test. You don't have to heat it or anything, you just put iodine on it, and it will go blue-black. The second test is for reducing sugars. Reducing sugar would be something like glucose, something like fructose. Pretty much all monosaccharides and most disaccharides are reducing sugars because they reduce ions such as copper. So copper ions are put into the mix in the form of something called the Benedict's reagent. It's a blue solution. So you add Benedict's reagent and then you heat. You will not get the marks if you do not write heat. Okay, don't put in water bath because a water bath could be at 20 degrees. Okay, there's water and it's in a bath, it's 20 degrees. It doesn't imply heat. You have to put heat. If there is reducing sugar present, it will go through a series of colour changes, but essentially it goes from blue to brick red. Blue to brick red. And then finally is the test for non-reducing sugars, namely sucrose. So sucrose will not show up on a reducing sugars test, it will just stay blue. So what you have to do is you have to add acid. Okay, you have to add acid. What that does, that breaks down the bonds in between sucrose molecule. You have to heat it as well. So you add acid and heat. That breaks down the glycosidic bond. You then have to neutralize the solution, usually by adding sodium hydroxide. And then you carry out the Benedict's test as you would with a reducing sugar. So that is everything you have to know about carbohydrates. Enjoy.